speaker is Dr. Shyam Aggarwal, Senior Consultant, Department of Medical Oncology, Sir Gangaram Hospital, New Delhi, India. The topic of the talk is HER2 new positive breast cancer, new developments.
Thank you very much, Professor Gopinath, for making us a part of this endeavor, and respected chairpersons. So I'm going to be talking about clinic as to what is happening in the field of HER2 positive breast cancer in the metastatic setting. Next slide, please. Can we move the slides? Okay. Okay. So if you look at the overview of HER2 positive breast cancer, so nearly 15 to 20% patients will be HER2 positive, which means that there's a gene overexpression which is determined either by immunohistochemistry or by FISH testing. And that actually confers a bad diagnosis to a patient who has breast cancer. At least that was the story, say about 20 or 30 years ago. So this is how you determine by immunohistochemistry as to whether the patient is HER2 positive or HER2 equivocal or HER2 negative, depending upon the degree of staining which you see on the membrane of the tumor cells. So this started changing in 1990s when the first drug called trastuzumab came out from Dennis Lehman lab at UCLA. And after that, you've seen several drugs which I'll be talking about in the next few years, such as pertuzumab, neratinib, TDM1, lapatinib. So a lot of water has gone, under the bridge, and so much of development has happened, as you will see. So I'm aware the next talk is on early breast cancer, so I'm not going to be talking about that. But trastuzumab changed the scenario in the adjuvant setting for patients with early breast cancer. And then, of course, came pertuzumab and TDM1, which I'm sure will be discussed in the next trial. I'm going to be focusing on metastatic HER2 positive breast cancer. So as I said, around 20% of these patients of breast cancer, we have to do it. It is a mandatory test today. And if you look at the curves from 2008, the curve for survival, overall survival, is going up with the availability of newer agents. And now if you see at 5 years, almost 50 to 60% patients with metastatic breast cancer can survive. Earlier, everybody was dying at nearly 1 to 2 years. So that's the kind of impact this research has made and the availability of these new drugs. So this is to show the mechanism of action for these various drugs. So you can see the cell membrane and then the antibodies which have been developed, because the trastuzumab, pertuzumab, and margetuximab are antibodies which are directed against HER2 gene. The three drugs mentioned down there, lapatinib, neratinib, tucatinib, work in the signal transduction pathways to inhibit the HER2 directed signal transduction pathways and stop the cell division. But what actually has happened in the last few years is on the other side of the slide, which tells you about the antibody drug conjugates, where you have the TDM1, you have the TDXD, and you have the trastuzumab ducharmazine. So they've really brought in a revolution. And I wish there is somebody here to understand the ADCs, as they are called, to really transform a patient who's suffering with metastatic breast cancer or even early breast cancer. If you have an effective ADC, things can change. I'll just show you. So we have several drugs now for MBC, metastatic breast cancer. How do you sequence? So what do you do first? What do you do second? What do you do third? So now I'll just showcase some of these studies. You see this Cleopatra study in which docetaxel, which is a chemo drug, was combined with trastuzumab and pertuzumab. And this was compared with, just the trastuzumab and docetaxel. And you see the curve, which is better for the dual inhibition of the HER2 because pertuzumab works through a HER2, HER3 pathway, trastuzumab works through HER2, and docetaxel is combined in first line. So if you look at the overall survival at almost, 90 months or 100 months, almost 30 to 40% patients are surviving. 1 to 2 years, everybody was dying. And now if you use this combination, which is readily available in our practice and we are doing it, so we expect almost 35% patients, 1/3 of the patients alive at 7 years. So that is the latest data which we have, and this is readily available in our clinic and we are doing it. So then what after the drug fails? As I said, 60–70% patients are going to fail. So then you have this TDM1, which I'll just show you, which was compared with the other drug combination, which is lapatinib and capecitabine. Little better, because the difference between the curves is a little, not too much. But then something has happened, as I said, through ADCs in the last 5 years or so. So this is the era of ADCs. This is the trastuzumab. Trastuzumab is an antibody against HER2, as you know. So this is combined with a topoisomerase inhibitor payload, and the ratio of the antibody to the drug is 8:1. So the TDM1, which I just showed you, we have the anti-microtubule payload there, and the ratio is 3.5:1. And this is a cleavable linker. So it's a story of linker, it's a story of payload, and of course, joining it with the antibody so as to deliver the payload to the tumor cell to maximize the toxicity of the drug into the tumor cell. And this is what TDXD has really done in the last few years. So I'll show you some data there. So as I said, the second line therapy after the failure of trastuzumab, pertuzumab, docetaxel was the TDM1, which is the red one, the trastuzumab emtansine, was compared with trastuzumab deruxtecan, which is the TDXD. And you see the outcome here? Sorry. So you see the difference in the curves. So the response rates in the second line are touching 70 to 80%. And if you look at the 50%, if you look at the hazard ratio, 0.33, which means that there is a 67% chance of not relapsing when you are using TDXD than as compared to TDM1. So therefore, TDXD is a better drug than TDM1, and it also has an overall survival benefit. So I think I'll just skip these slides because we're running short of time. The point is to state that it works all through all types of baseline characteristics of the patient. So I'll skip these slides in the interest of time. So the point I'm trying to make is that TDM1 now is going. TDXD is the in thing, which is the deruxtecan. So this is the other drug. So the tucatinib and lapatinib and neratinib, these are three drugs which work inside the cell, and they inhibit the signal transduction pathways, which are started with the trastuzumab. So then you have the tucatinib, trastuzumab, and capecitabine combination when compared with trastuzumab and capecitabine. And again, this is another combination which is showing you, a better outcome in terms of PFS. Not perhaps that good as TDXD, but comparable. So I'll just skip in the interest of time. So again, you see the overall survival is better for tucatinib combination, which is a intracellular drug inhibitor of the HER2 pathway. So I'll skip these in the interest of time. I'll not go into the safety. So then people thought that we have two drugs. You have tucatinib, which works in the cell. We have TDM1, which acts on the surface of the cell. Let's combine them and see whether it works better than TDM1. Well, it does, but the difference is not much. Of course, you do have some brain, efficacy with tucatinib, which I'll show you subsequently. So I think this is the way we are, moving from trastuzumab, pertuzumab, then you have the TDXD, and you have tucatinib. So 4 drugs. So so what after that? So neratinib is another drug which works inside the cell, and there are data to support the use of neratinib, especially so in patients with ER-positive breast cancer, early breast cancer, and also in the metastatic disease. But as you see, the curves are not very much separated, and therefore it's not very great to get neratinib, especially when you have the diarrhea as a toxicity. There's some efficacy in the CNS, which I'll be just coming to. So this is the 5th or the 6th drug, which is the margetuximab, which again, activates through the Fc engineering pathway, the immune responses. And if you look at the curves there, not great, but then little better than the, trastuzumab and chemotherapy than you compare with margetuximab. So the point is, there's a plethora of drugs targeting HER2-neu gene, which was supposed to be a very bad disease to begin with. Okay, so there are some side effects. I'll not go into the details of this. Nausea, vomiting, I think we as chemotherapists, are used to that. So TDXD does cause that, and we have to take care of that with the good drugs. But I think the most important side effect is the interstitial lung disease, which is associated with the TDXD, and we are supposed to do CT scans to identify that and stop the drug or initiate steroids to prevent the toxicity from being a killer. Almost happens in about 10% of the patients. So I'll skip these slides in the interest of time. So there are several trials going on, which I'll just come to. So this is the, so once we find a drug which is available to us in second line now, so people start moving it in different combinations in the first line, as you see here. So, you want to defeat now docetaxel, pertuzumab, and trastuzumab. And if you see here, the trial says you can use TDXD with, with pertuzumab and see whether it works better than the previous combination. So that data is yet to come. The trials are ongoing at this point in time. So now there are, I showed you in the first slide, about 5% patients have HER2 along with the HR overexpression, which is the ER overexpression. And there's a crosstalk between ER and HER2. So now people are trying to, and these are the preclinical studies, so people are now trying to look at the combination of trastuzumab and abemaciclib and fulvestrant, and showing you the efficacy of these drugs in the MONARCH-HER study. Little difference here, but then I think it may be meaningful over the next few years as the studies evolve. So there are several studies which people are doing with the CDK4/6 inhibitors along with anti-HER2, but the data is yet to come in. So this is another very important issue, which is the brain metastasis. Nearly 50% of patients of HER2 positive disease will have brain metastasis at some point of time in their journey of cancer. And the problem is trastuzumab, pertuzumab do not cross the blood-brain barrier, neither do the chemotherapy. But look at the TDXD data. I'll just show you this. So nearly 40 to 50% patients are benefiting with the TDXD when you combine with the chemotherapy. So this is an effective agent. So I'll just, I'm skipping these slides. So you see that curve, which is so very different, the green one and the red one in favor of TDXD. So I'll skip these and look at the trial which is going on now, which people are looking at. But you have a drug now which crosses the blood-brain barrier and help patients with brain metastasis. So neratinib, I've already mentioned about, they're combining with TDM1. So I'll just skip these in the interest of time. So the tucatinib data which I showed you, again, is a drug which crosses the blood-brain barrier, and you can see the overall survival benefit then as compared to patients who are not on tucatinib. So I'll just skip these. There are several trials going on, and then this really brings us to a very important point, that the TDXD is so effective that patients who have very low expression of HER2-neu on their surface, well, they can also benefit from it. And this has really brought in 60 to 70% of these breast cancer patients who have HER2 low expression will benefit. So as I said, 15–20% are HER2-neu positive, but then the rest are either HER2 zero, almost 60% are HER2 low. So this is a huge, huge progress in the last 3 to 4 years. Look at this data for patients who are HER2 low, and when you use the TDXD, see the curves separating. So this has given us an added dimension to the treatment of patients where HER2-neu is either 1+ or 2+, but the FISH is negative. So therefore, for all these patients, we have to keep our minds open, because we forget about HER2 low. We see HER2 1 negative, HER2 2 positive, but FISH negative as negative. But then you have this drug available now, which can change the scenario of a given patient. See the difference in the curves and the hazard ratios. So I just wanted to make this point that the HER2 low can benefit from TDXD. So I think there are several new drugs now which are coming in. I'll just skip these slides because we are running late here. There are several other ADCs, which you can see. So basically, trastuzumab is combined with vedotin or certain, this is a list of drugs here where, you're looking at HER2 bispecific antibodies. I have little data there, but I'll just skip this. So I think the ADCs are going in a big way for management of not only breast cancer, but a variety of cancers. So, we have drugs like amivantamab and things like that, and ADCs is the way to go. There are almost 30 to 40 ADCs either approved or in trials for a variety of cancers. So I'll just go to the last slide. And this is the treatment algorithm as it exists today, but it might change in the next few years because TDM perhaps could become as a first-line treatment. So currently, at this time, taxane, trastuzumab, pertuzumab in metastatic breast cancer is the first line. The second line, I think people want to use the trastuzumab deruxtecan, irrespective of CNS involvement, but some people might want to use tucatinib. After the second line failure, well, then you have tucatinib or TDM1, and then you do have other drugs like margetuximab, neratinib, and lapatinib. So I think we've come a long way for HER2-neu positive disease in the metastatic setting because of the availability of these newer agents, and we are able to transform the lives not only in terms of longevity, but also the quality of life. So with this, thank you very much.